Hello students, in this video we'll be continuing with the topic of test of significance. Well, as we have discussed that researchers employ hypothesis formulation, that means researchers basically formulate hypotheses or assumptions which can be tested, and based on those assumptions after being tested we can arrive at certain results through which we can figure out the interpretations, conclusions, and implications for n number of things. But for testing these hypotheses, what we have to do? Well, for testing the hypothesis, the test of significance has to be conducted. The test of significance may involve many methods or different statistical techniques, but there have to be certain criteria or there has to be certain decision making involved so as to select the right one. So in this video, first of all, we will be discussing about what is the test of significance, what are the terminologies associated with it, and then we'll be also discussing about the different test of significances or different statistical tests which can be conducted for better understanding and for obtaining better results. Now, to start with the introduction, all scientists or researchers look for the answers to following question. Now, any kind of research is being there, whether it can be scientific research, social science research, it can be a research associated with some historical fact, any kind of research or any kind of scientific inquiry it may happen. But the basic questions of the researcher or scientist are how probable the difference between observed and expected result is by chance only. Because there are some things which are already expected before conducting the research. Before any researcher or scientist starts the research, they have some expectations about the results or some expectations about the phenomena. And then there are the observed values which come after running the scientific inquiry or after conducting the research. So after doing the research, you get some results or some observed values. So you have to see that what are those differences, why these differences are there between the probable value or the expected value and the observed value, and are these differences by chance only or are there some other factors which are responsible for such differences. So this has to be known. And another question about the differences between these things is the difference statistically significant or not? This is a very strong question and very important to answer, that whether these differences which are they statistically significant or not. So sometimes differences can be very small or differences can be so negligible that they are not statistically significant. They are not significant to note them. So the differences can be there, the differences can be statistically significant as well as differences can not be statistically significant. So these are the questions which basically roam around the mind of the researchers or scientists when they are conducting the study and after they conduct the study. So to answer these type of questions, the test of significances has to be conducted. So the differences which are observed can be noted out whether they are significant or not. And another thing, are these differences by chance only or are they deemed to happen? So let's continue with our discussion of test of significance. So test of significance is a formal procedure for comparing observed data with a claim, that is expectation or hypothesis, whose truth we want to access. So in very easy language, in basic language, you can say test of significance is nothing but it is a procedure, it's a formal process for comparing our observed values data with the hypothesized or expected values. Alright, so we are just comparing those values, we are just comparing the results with the expectations to see whether these results or these differences are significant or not. So So the test of significance is used to test a claim about an unknown population parameter. So it is a test to, it is basically procedure to test a claim, to test a thing which has been acclaimed by the researcher about an unknown population parameter. So the population parameter which is not known to us, we are inquiring about it, we are knowing it, so we want to test the claim about it, whether our claim is true or not. A statistical test uses data to evaluate a hypothesis by comparing sample point estimates of parameters to values predicted by the hypothesis. So basically, in technical language, this definition can be there, but for understanding point of view, you can look on the first point on your screen, which says that it's a formal procedure for comparing observed data with the expected one. So the level of significance. Now what happens whenever these tests of significances are run, whenever we use data to analyze the hypothesis and come out with a solution that the null hypothesis or the statement is true or not, we come out with the results that the test of significance says that the sample points which we are expecting are not true. So whenever we are doing these things, we have to see the p-value. So the p-value or level of significance has to be considered very carefully, because it may differ from research to research. It may differ from my study to your study. You may get something else, I may select something else, because the level of significance is largely based upon your sample size and also upon the accuracy that you need. The nature of research also dictates what level of significance there can be. But what is this level of significance? Well, we can say that level of significance or significance level is the maximum value of probability of rejecting null hypothesis when it is true. So simple terms, in very easy language, you will be struggling a lot to find this kind of simple statement defining level of significance. So we can say that it is the maximum probability level of significance, whichever you are selecting for your research, it is the maximum at most probability that you are going to reject null hypothesis where it is true. That means you are going to commit type 1 error. So significance level is the maximum probability of possibility that you are going to commit the type 1 error. So p-value is a function of the observed sample results that is used for testing a statistical hypothesis. So it is a function, it is a value, it is a figure, percentage, which you are using, alright, for observed sample results. Whatever observed sample results you have got, so you are using that for testing the statistical hypothesis. Or in another simple language, you can say it is the percentage of risk that the researcher takes. So whatever results you are getting, what amount of percentage of possibility is there that you are interpreting it wrongly, it is can be understood as level of significance. So generally, if you see researches around, you will find that normally p-value or significance level is taken as 5%, that means 0.05. So 5% significance means that you are taking 5% risk that your null hypothesis is true, but you are rejecting it. Or in other words, you can say that there is 95% confidence interval that your results will lie within the range that you are specifying. So if you are taking 5% significance, that means p-value of less than 0.05, it means the result is significant and the standard error is 1.96. In other words, we can say that 95% confidence interval is there. But suppose you are taking 1% significance, that means 0.01 is your p-value. The results are highly significant in this, and the confidence interval is 99% here. However, when researches are carried on for a very specific and accurate task, suppose some vaccine has to be created, so their accuracy is needed at a very high level. Very high accuracy is needed. So we'll be taking significance value of 0.1%. So we can take only 0.1% risk that we are giving out, we are interpreting our results wrongly. So 0.1% significance means p value of 0.001, which is highly significant. And the confidence interval here is 99.9%. That means our results are 99.9% accurate. Now, in our earlier video also we have discussed about the steps for hypothesis testing. Now, those were the theoretical steps, those were the steps which are taken up. Those are not wrong. I'm not saying those are wrong. Those are the theoretical steps which you can write very easily and with very confident. They are not wrong, they are correct to the utmost accuracy. But here I am giving you some steps of hypothesis testing through test of significance. Those are valid in every condition. These are valid only when test of significance are to be conducted, and in practical terms always test of significance will be there whenever there is hypothesis testing. There are some qualitative studies being taken up for hypothesis testing also, but there also test of significance has to be there. Different methods of testing out the qualitative data are also developed. So these are the steps which you are going to follow for test of significance or hypothesis testing. When you are asked in your examination, you can either write those or write these. None of these process is wrong, but this one is more of a practical one. So first step is defining the research question. You first have to define the research question. Whenever you are conducting any research, you start your research with a research question. So the process of hypothesis testing starts with the very thought of research question. When you are framing or when you are articulating the research question, that is the point, that is the spot when you are actually conceptualizing that you are going to test a hypothesis. And after defining that research question, you are stating the hypothesis. You have to state the clear-cut, concise hypothesis in a very clear manner, in a very clear statement. Either the null hypothesis or alternate hypothesis or both of them, you have to state them in a very clear-cut, concise statement, which can be understood by people at mass, and which is very easy to understand, which is not ambiguous. The third step is selecting appropriate test. Now, how to select appropriate test? We'll come to that in the later slides, because we are going to discuss about the common tests which are used. So what test you are going to use depends upon the relationship which you are defining in your hypothesis test. Now many times people ask that, sir, how do we determine which test we have to use? We have collected our data, but now we are stuck into the appropriate test. Now friends, you are wrong when you are collecting the data and then deciding which test you want to apply. Your thought of selecting the right test depends upon the hypothesis which you have defined. Then at a later stage, it depends upon the type of data you have. But first, the foundation of appropriate test depends upon the hypothesis which you have framed, the relationship which you have defined. Suppose you have defined the relationship which is impactful, which is a causal relationship. There a regression would be needed. Suppose your hypothesis talks about relationship between the two sample means. So there you have the choice to be made from comparison of mean test which can be t-test, z-test, which can be ANOVA, depending upon the type of data. But depending on the relationship, you are straightforwardly shortlisting the test which you have to take. So selecting the appropriate test is the next step which you have to take, and then you have to calculate the test criteria. Now you have collected the data, you know the test which have to be applied, then you apply the test and you calculate the test criteria. You calculate the value. Suppose you are running t-test, so you calculate the t value, which is the test criteria, and you select the significance level which you have to take for your study, and then you compare the test value with table value and check the value of p. So after calculation the test criteria, you calculate what value of test you are getting. You decide the acceptable level of significance, that is p value, usually 5% is taken, and then you compare the test value with table value. Suppose you have got t value at certain degree of freedom and your significance is 5%. So you observe the table then, the whether my value which I have got, the t value, whether it is in acceptable range or in non-acceptable range. And then you check the value of p because p value tells you the significance. Then you will accept or reject the null hypothesis, or you will accept or reject the alternative hypothesis based on the value you get. So suppose your p value is less than 0.05, that means your results are statistically significant, and you are going to reject the null hypothesis in case your p value is less than 0.05 when you are taking 5% level of significance. Then you have to take out an inference, you have to do interpretations of your result, you have to give conclusions and implications, and this is how a research ends, or this is what a research has to provide, these inferences. Now starting with the test of significances, we are going to first discuss the t-test, which is very commonly used. So the topic or the theme which t-test talks about is the differences between means or proportions of two groups. Now these groups can be independent groups, similar group at different times, or there can be different groups which are being tested. We will be coming to that. So first of all, let us understand that t-test is a part of inferential statistics. What is inferential statistics? The statistics which is used for taking out inferences. The statistics which is used for testing the sample, taking out the results and inferring that results to the population. So t-test is a part of inferential statistics, and it is conducted to determine whether there is a significant difference between the means of two groups or not. So you are basically concerning with the differences between the means of two groups. The mean value of one group, is it different from another group or not? The behavior of one group, the mean behavior of one group, is it different from another group or not? The choice behavior of male is different from female or not? The employees who are selected through online portal are having the same mean response or different mean response from the employees which are selected from offline portals. So through t-test you are basically comparing two groups with the help of their means or with the help of their proportions. So depending on the nature of sample, now depending on the nature of the groups which you are testing, there can be different type of t-tests which are applied. So the nature of sampling can be two different groups or one group at different time. So suppose the first type of t-test is paired sample t-test. So paired sample t-test is applied when your sample is single, and that single sample is observed or has been taken responses at two different times. So your sample is paired. Your sample, the two groups, the two sample means which you are having is paired to one group only. Suppose you want to check the behavior of employees before training and after training. So this is a paired sample. The same sample you are using at two different time horizons. So what you are doing, you have to conduct a paired sample t-test in that case. So the two sample means which you are comparing are paired to each other. That means that both sample means are coming from a single group. In that case, you are going to apply paired sample t-test. However, when we talk about independent t-test, independent sample t-test means the two groups or the two samples which you are going to compare are independent of each other, are different from each other. They are two different categories. So the example can be when we are basically checking the differences between male and female, or we are checking differences between the migrant workers and the native workers. So what we are doing is we are comparing two different groups, two independent groups. We are comparing two independent samples with each other. So the mean which is there, the mean values which you are comparing are belonging to two independent samples. In that case, you have to apply the independent sample t-test. There can be two type of independent t-test. One is equal variance and another one is unequal variance. Now these are applied when you know the sample variance. Suppose the two groups which you are comparing, they have equal variance. Both the samples have equal variance. In that case, equal variance t-test is applied. However, when you know that unequal variance is there, in that case, you apply unequal variance independent t-test. The example of relationships which are or hypotheses which are tested using t-test is, suppose there is a hypothesis to test whether the behavior of males differ from females while buying something online. So if your research question or hypothesis is talking about such a behavior, such a relationship, you are going to conduct a t-test. And this is independent sample t-test because male and female both are independent of each other. Now in another example, suppose you want to check whether employees' performance is different after training. So that means you want to check the mean of their performance before training and after training. So the two samples which you are collecting is from same group. So this is a case of paired sample t-test. Likewise, for any relationship which you are going to check or any hypothesis which you want to test, you are going to see whether the relationship which you are going to test is this kind of relationship. In that case, we are going to apply t-test. Now there are certain assumptions which have to be there before conducting a t-test, and usually one thing more which was left in previous slide, actually that was not written as a bullet. T-tests are usually applied when we are concerning about a smaller sample size, because when we are concerning with a larger sample size, we apply z-test. We'll keep coming to z-test in the next slide, but first let's understand the assumptions which are there for t-test. So before conducting a t-test, you have to ensure that your sample is randomly drawn. Out of the population, you have taken sample randomly from almost whole population. The second assumption that has to be fulfilled is that the normal distribution of population has to be there. Now this normal distribution of population is ensured by normal distribution of sample. So when you are taking a normal distribution sample, or when you are ensuring that your sample is normally distributed through random sampling, so you presume that there is normal distribution of your population. However, if this thing is checked first and foremostly, it is very good because the results may be distorted. The third assumption is dependent variables on interval or ratio scale. Well, when we are talking about these variables, so the variables which are there on this t-test, the variables which we are using as a classification variable or the dependent variable, they should be on interval or ratio scale. The groups have almost equal variance. This has to be ensured when you are applying the equal variance t-test. The next one is z-test. Well, z-test is also an inferential statistics, and it is based on the normal population distribution. This test is usually done on a larger sample than a t-test. The aim of both the tests is almost same, that it is also used for comparing mean of a sample to some hypothesized mean for the population in case of large sample. So basically you are comparing mean of one sample with another sample, or what you are doing, you are comparing the mean of the sample to test whether it is same as hypothesized mean or not. An example could be to check whether the accuracy in the result, or suppose there is a production going on for nuts and bolts. So suppose you want to check what is the quality assurance. So suppose you fixed up the mean that quality assurance mean would be 9.5 out of 10. So your hypothesized mean is 9.5. So you want to check whether the actual mean is same or statistically different from our hypothesized mean. You have to conduct a z-test in such a situation. Z-test can also be conducted when you don't know your population variance. You don't know how much your population is varying. You don't know how much your population variance is. In that case also z-test has to be applied. Another example of a relationship where z-test is applied can be, suppose you have hypothesized or you have expected that the result, average result of the college or the average performance of students would be 90%. So you have a hypothesized mean which is 90. So you will see the observed mean, suppose it has come out to be 87. So is this different statistically significant or is this different non-significant? To test that you can use a z-test. Z-test can also be used for comparing the means of two independent samples. Suppose you want to see whether the performance of male employees or male students is statistically different from the performance of female students or female employees. So the two independent samples can be tested using z-test. However, paired samples are usually not tested using z-test. However, it is possible to test them, but usually z-test is not used for testing the paired samples. It is usually used for testing the independent samples only. So it can also be used to judge significance of difference between means of two samples. As I just said, that there can be two groups whose means can be compared. How it is done? The z value is worked out or calculated and compared with the table value at a specific significance level, and the hypothesis is then accepted or rejected accordingly. The assumptions for z-test are this: the sample should be drawn randomly, the data should be normally distributed. Now it is considered that when you are considering a large sample, the data comes usually as normally distributed data. So a large sample is the third assumption for z-test, and this large sample means a greater than 30 sample. The next one is f-test. Well, f-test is used to compare the variance of two independent samples. In t-test and in z-test, we were comparing the means of two samples, but in f-test we are comparing the variances of two samples. It can also be used in the context with ANOVA for judging significance of more than two sample means at one and the same time. So for with ANOVA also it can be used for checking the variance first, because in t-test also sometimes people check the f value before conducting t-test just to ensure whether the variances are equal or not if they want to apply the equal variance t-test. To ensure whether the variances of two groups or samples are equal or not is the basic thing of f-test. Coming towards cross tabulations or crosstabs. Now what happens whenever we are conducting any research? We are dealing with certain variables. Alright, there are certain variables which we are dealing with, and there are certain frequencies or certain correlations which are there between the variables. So what is crosstabs? Crosstabs is a method, or it is a technique used for presenting the two variables in a cross tabular manner, where the variables are represented through different rows and columns, and you can apply some basic test on it. So it helps to take informed decisions by identifying the patterns, trends and correlation between parameters of the study. So you can basically study the patterns or trends which are there within your variables, or the correlation within your variables. It displays the joint as well as relative frequencies of two categorical variables. So two categorical variables which are measured on nominal or ordinal scale, they can be jointly or relatively presented in a cross tabular manner. It represents data in a two-way table and provides a variety of tests to be conducted in that form, mostly for comparisons and categorizations. So mostly when we do cross tabs, when we present our variables using cross tabular manner, we are presenting it, or we are using it for a better comparison or a categorization. So you can categorize the values or groups on the basis of the need of their research. You can also compare between two different classes of two different variables. The important values which can be calculated out of crosstabs is you can calculate the correlation between variables, you can know the phi and Kramer's value, you can also bring out the chi-square and other things using the cross tabulation. Now coming towards chi-square test. Chi-square is a very important test which is conducted in many researches. It can be done for parametric as well as non-parametric data. Well, just for a sake of understanding, parametric data is when the parameters are defined, when the data parameters are known, the mean, standard deviation, the different frequencies and the mathematical operations are there in the data. And when you are not knowing the different parameters of the data, when you cannot work out the different parameters of the data, it is termed as non-parametric data. So for parametric way, chi-square is used for comparing variance with theoretical variance. So whatever variance you are expecting and whatever variance is actually existing, you can basically test that variable, that variance using chi-square test. The obtained variance can be compared to expected variance, as I just said. We can also use it to judge whether the random sample is drawn from normal population or not. So suppose you have drawn a random sample, you have got its data, you want to know whether this data is coming from a normal population or not. So in that case also chi-square value can be used. Talking about non-parametric way, So as a non-parametric test, it can be used to test the independency or dependency of a variable on another variable. So you can know whether a variable is statistically related or statistically associated with another variable or not. Suppose you want to know whether the learning is statistically associated with presence, with physical presence or not. That means you are going to check whether online education is effective or not. So you want to know whether the presence, physical presence is associated with learning or not. So in that case, you can use the chi-square non-parametric test. Whether you want to know that your selection of a certain product is associated with your gender or not. In that case, you can use a chi-square test of independency or chi-square test of association. So it can check whether the categorical variables are dependent or independent upon the other variables. It can be used as a test of goodness of fit also. That means it is used as a test of goodness of fit. Whether sometimes what you have to do is you have to define a theoretical model. You have to fit data in it. So you have to check whether this theoretical or conceptualized model is actually a goodness of fit or not. Whether these concepts which you are aligning with each other are aligning with each other theoretically or not. So you can use a chi-square on that data, and in this case, if calculated value, the resultant value is less than the table value, you take it as a goodness of fit. However, in other cases, you have to see the calculated value has to be greater than table value. But when you are running a goodness of fit test, you have to see the calculated value has to be less than the table value. The assumptions is random observation should be there. The data has to be collected randomly. All items in sample must be independent. The sample elements or the sample units should be independent from each other. No sample unit should be dependent upon another sample unit. Large samples are usually considered when we are doing chi-square, and a small sample chi-square may not produce correct results. So a sample greater than 50 is considered. And when you are considering about the non-parametric chi-square test, which is run for independency or association, the variables must be on nominal or ordinal scale. Coming on the next test of significance, which is commonly conducted, ANOVA. Well, ANOVA stands for analysis of variance, and as we just discussed that t-test and z-test are used for testing differences in samples of two groups only. So we had a detailed discussion upon t-test and z-test, that they can test the significance of difference between two sample means. Those means can be independent or those means can be paired, but there have to be two groups only. But what if there are more than two groups? Right? You were testing the significance of difference between behavior of two groups: migrant workers, native workers. Suppose there is some third category which is given into it as the migrant native skilled workers. Suppose there can be three groups of gender: male, female, transgender. So in that case, how will you apply t-test or z-test? You cannot apply that. For when there are more than two groups, when the sample means which you have to test belongs to more than two groups, then ANOVA has to be applied. So it enables to test for more than two sample means, or more than two groups can be compared using an ANOVA. So using this technique, you can draw inferences about whether the samples have been drawn from population having the same mean or not. Again, the samples which are drawn, you can check whether these samples belong to the same population or not. This is also done using ANOVA. Suppose you have some data, 3 groups of data. So you want to check whether these 3 groups of data are having the same mean or not. The population from which these 3 means are taken out, those original population were same or not, you can test using ANOVA. It is important in the context of all those situations when we want to compare more than 2 populations, such as in comparing the yield of crop from several varieties of seeds. Suppose there are 5 types of seeds are available. You want to check which type of seed is giving you more yield, which is more productive. In that case, ANOVA is very helpful. The gasoline mileage of 4 automobile. There are 4 automobiles, there are 4 cars. Suppose there is Wagoner, there is Santro, there is Nissan Micra, and another one is Maruti Swift. You want to compare the mileage of these 4 cars. So the sample mean of 4 different groups are available. ANOVA can be used. The learning habits of 5 group of university students, and so on. So whenever there are more than 2 groups whose differences have to be seen, ANOVA comes into the picture. The basic principle of ANOVA is to test the difference among the means of populations by examining the amount of variation within each of those samples. So how does ANOVA work is, ANOVA tests the means between the populations by examining the amount of variation within each group. How much variation is there within each group relative to the amount of variation between the samples. So it is more accurate because it is conducting a more rigorous research. The assumptions of ANOVA, these are very important. If these assumptions are not fulfilled, or if these assumptions of ANOVA are not taken care of, the results may not be correct. Sometimes the results may not be produced also. So each group sample is drawn from normally distributed population. Is very important that whatever group you are taking, whatever data you are drawing from the population, those populations should be normally distributed. There is the concept of homoscedasticity. What is homoscedasticity? Homoscedasticity says that all population must have common equal variance. So the populations from which you are collecting your sample, from which you are collecting your data, those different populations should have equal variances. Now this condition may be fulfilled when a large population is there of each group. So this condition is presumed to be fulfilled. However, for testing the homoscedasticity also, you have to see the relevant values. All samples are drawn independently. Very important. The samples which you are comparing with each other, those samples should be drawn independently. Those should not be paired with each other. Then within each group or within each sample, all observation or units should be independent. Each group also, every unit, every element should be independent of each other. It is also an important assumption of ANOVA. Well, thank you so much for your